begin by just a very sort of brief preview, which I'm happy to talk about more during the Q&A at the end, by pointing out that there's a big disconnect between how your brain looks and who you are. So, and in particular, in relation to some very unusual looking brains. Then the main focus is really very recent work. None of these have appeared yet, but they will appear soon in print, 2 papers that try to predict individual differences in personality and in intelligence from looking at your resting state functional brain activation. And then actually leave quite a bit of time for sketching what I think are interesting future directions that this work is going to go in. And I should say, throughout, if you have a pressing question or if something is unclear, please just interrupt. So, unusual looking brains. This slide is actually one of the first slides I use in the introductory neuroscience course that I teach at Caltech, to make the point that it's extremely difficult, indeed frustrating, to hold a post-mortem human brain and to realize that this generated a person and a world and behavior and cognition, and you really can't tell anything about it. I mean, you can look at that thing as much as you like, and here's a healthy human brain. This is the brain of Albert Einstein. This is the brain of a schizophrenic person. I noticed that these brains are in worse shape than the beautiful brains, post-mortem brains that you're collecting here at the Mind Institute that Cindy Schoon was showing me earlier today, which don't have these nicks in them. Unless you take 2 or 3 or 4 looks, you might even think that this is a human brain. It's actually the brain of a chimpanzee that's been scaled up to the same size. So it's very difficult to tell anything. You can't tell if somebody's male or female or has schizophrenia or autism just from looking at a brain. And indeed, there's a big disconnect. So as you probably know, in some developmental disorders, in this case congenital hydrocephalus from a paper in the New England Journal of Medicine, you can have cases, rare ones, where in a developmental context, a brain like this, that's only about 10% of a healthy adult human brain, generates a largely normal individual, not somebody without problems, but a person who has a job and a family. So the disconnect is a big disconnect between how your brain looks or even how much brain you have and who you really are. In my lab, just very quickly, we've been studying and are still studying 2 really fascinating populations that emphasize that point. One are patients who are born without a corpus callosum. So they're like the split brain patients that Roger Sperry studied once upon a time, only congenitally so. If you take a healthy brain here on the left and you carve away the top, you can look down at this bundle of 200 million fibers, axons, that connect the left and the right cerebral hemispheres normally, but are absent in patients with agenesis of the corpus callosum. So here is a MRI scan along the midline, a sagittal MRI, where in a healthy brain, you see this bundle of axons, the biggest white matter tract in the brain, the corpus callosum, cut in cross-section, which is missing, and there's also lots of misfolding along the midline in the brain of a patient with agenesis of the corpus callosum. So a fascinating disorder, and here's a diffusion imaging scan in a healthy brain. These colors encode the directions in which connections in the brain run. This big red thing here is connections that go from left to right, the corpus callosum in a healthy brain, missing in the brain of a patient with agenesis of the corpus callosum. What happens instead is that these fibers that would normally cross the midline instead make abnormal connections and abnormal white matter bundles within each hemisphere. So, it would be as if I cut my laptop in half and rewired a whole bunch of stuff. It wouldn't work at all. These brains work remarkably well. In fact, when we image these adults, not only do they have families and jobs, IQ in the normal range, very subtle deficits that present more or less like high-functioning autism, but in fact, if you do functional imaging, not structural imaging like is shown here, you find that functional connectivity, functional networks in the brain look entirely normal. And we have no idea how that arises in development. So it can't be the case that just the basic structure of the brain determines function and cognition and what kind of person you are. It's much more complicated than that. The other population, just quickly to highlight that we're looking at on a shoestring, are a few patients where as children, because of epilepsy, a neurosurgeon resected half the brain, patients with hemispherectomy. And it goes all the way down, and you have cases where really half the brain, including diencephalon, amygdala are gone, the person will walk in and talk to you and tell you about their life. They have difficulties, but again, half a brain doesn't equal half a person. And we don't know at all, again, in a developmental context, how it is that half a brain structure can generate a largely normal, remarkably compensated cognition and behavior. So I point these cases out. They're sort of fascinating side projects in the lab, but they emphasize to us 2 points shown here, that if you just look at a brain, indeed, you can't tell much about a person from looking at their brain. With your eyes, you can tell, and that's the rest of the talk, something about a person from looking at their brain with the tools of neuroscience. And so we do need neuroscience to really look and understand brain function to say anything about cognition and behavior. Just looking at it from the outside, as you've seen by these examples, is only very, very loosely related. And then the second point is that everything I'm going to tell you in the next part of the talk is all about healthy, typically developed adults, and the question of what can we predict about individual differences from those, when we know from the examples I just showed you and from the examples of developmental disorders more generally, that in a developmental context, there is remarkable compensation. And so ultimately, we need to understand not how functioning in your brain makes you the person you are, but how that came to be. So for the main talk then, can we predict from neuroscience data what kind of a person you are, who you are? And so when we started this maybe 2 years ago, we started looking at this question. And the first question we had was, well, what do you mean by a person? And the first order, with respect to the data we had, the first order answer would be it's everything you can measure about their behavior. That makes you who you are. You are what you do in life. And so the way that, for instance, in this data set, whose data are shown here, this is data from the Human Connectome project. So it's a big database of about 1,000 people that have been scanned. So we have MRI information. And they've given them a whole battery of tasks that sort of assesses what kind of person they are. Personality questionnaires that measure personality traits, lots of cognitive tasks that measure their ability for attention and memory and perception, lots of questionnaires about mood, negative affect, and positive affect. And when you do that, and you look across all these people, you find a pattern that's shown here. This is all the correlations between these different measures in 1,000 people. So red means that these things are positively correlated. So cognitive tasks are positively intercorrelated with one another. Blue means that things are negatively correlated. So all the negative affect questionnaires are positively correlated amongst one another. If you're anxious, you tend to also be depressed and endorse negative mood more generally, et cetera, et cetera. And they're negatively correlated with positive affect questionnaires. If you endorse being depressed, you typically don't say you're also happy. And there's more subtle ones. For instance, you can see that this may be hard for you to see, but if you endorse negative affect, if you're slightly depressed, you tend not to do so well on the cognitive tasks, whereas if you're in a happy mood, you do somewhat better. So there's structure amongst these, showing us that these things interact in some way. And ultimately, what we would want to derive from this is the kind of boxes and arrows pictures that psychologists have had all along, only informed by much more data and in a more principled way. That is, we would have all these different measures, all the different tasks that are listed here. Those are all these measures here that we can actually measure. And these have lots of questions associated with them. They may be very messy measures, very invalid measures. They have error associated with them. But from many of these, then, we would infer these latent variables: attention, memory, personality, intelligence, anxiety. And we would think that these would be connected with one another in that they have some influence on each other. So for instance, you might think a certain personality would predispose you to be in a certain mood, which has an influence on how you perform these cognitive tasks, something like that. So those are the behavioral data that we have to work with. And then what can we measure from the brain? Well, one can, and people do, measure everything they can. So you could think of, let's just find a biomarker that predicts personality or intelligence, so I can measure everything. And so people do measure lots of different things, as shown in this example paper here. And you could think of this as the sort of precursor to what will soon be the case when you go to your doctor. In addition to getting a blood test, you're going to get a brain scan, and personalized medicine will make use of the data that they obtain from that brain scan in order to predict things about your risk for certain disease, the most appropriate medication for you, and so forth. And so in this particular paper, they did this for just 10 subjects. They collected tons and tons of data, and so you get a big matrix for everybody of all their demographic information, who they are, how they scored on these different tasks, and you get lots of brain imaging data. So we and others have looked at this, and if you look at the literature, you find that brain structure, brain connectivity, lots and lots of measures about the brain have been purported, indeed, to predict lots of things about you, your intelligence, your personality, your mood, your risk for various diseases. So we focused on one of these measures, which is the functional connectivity between brain regions, this particular one shown down here, for the reason that I mentioned at the outset. If you just look at structure, it's very loosely related to who you are, and there's lots of things that happen between just the structure of your brain and your cognition and behavior. The actual proximal cause of your personality, your intelligence, your performance on task has to be brain function. And so functional data is what we looked at. Let me just give you a very quick, and you can take a break if you like. None of the rest of the talk depends on this, overview of the particular brain data that we collect, which is very common nowadays. And it's present in lots of big databases. It's called resting state functional connectivity. You just put someone in an fMRI scanner and you just measure activation all over their brain. That's it. And so what you get from that is activation at all different places in the brain at the same time. So I measure activation here at this red place in your brain. There's some signal. I measure your activation while you're just lying there in the scanner, not doing any task, at this blue region, and that's some signal. And I correlate all of those together. And what I get is a picture that looks sort of like this. So I take all these different places in the brain. Typically, we parcellate the brain into a couple of hundred or so regions. And what this shows here is a resting state functional connectivity matrix. It's very similar. In fact, it's depicted the same way, more or less, as the picture that I just showed you from the psychological tasks, where we had all the red and blue patterns. It's the same thing here. You get red and blue patterns. Here, what we've done is all the different brain regions are all the different rows here, and all the different columns are all the different brain regions. So the y-axis is the same as the x-axis, and it just orders all these different brain regions. So if I want to ask, I have a region in the left hemisphere, and I want to ask how well is it correlated with another region in the left hemisphere, I can find all these pairwise regions here. One thing that you'll notice right away, like with the picture for the psychological tasks, is that there's some pattern in here. It's not totally random. I mean, it looks complicated. But one thing you'll see is there's a big diagonal here. That just means that each region is pretty correlated with itself, or regions near it. So if I'm right here, my brain signal is going to be kind of similar to where I'm right here. That's not too surprising. A lot of it has to do with the resolution of fMRI. What's more interesting is this diagonal. This means that if I have a place in the right hemisphere, it's correlated with a corresponding place in the left hemisphere. This diagonal is the correlation between all the regions in the left and their counterpart in the right hemisphere, exactly all of those regions that would be connected by the corpus callosum, the big white matter bundle that's missing in those patients with agenesis of the corpus callosum I mentioned right at the beginning. So the same region, motor cortex on the left, motor cortex on the right, are correlated, and that's what this diagonal means. So the point is that you get some kind of relatively stable structure. This shows you which regions of your brain, if you're not doing anything, on average, tend to be activated together or not activated together. This is like a fingerprint, it turns out. So if I collect this from you in about 10 minutes or so these days, I can find you amongst 1,000 other people by just imaging your brain again. So it's like a fingerprint. It can be used to re-identify you, specific to a person. So it's something about how your brain works that's represented in this. What we then do is complicated, but the essence of it is shown here in this little schematic. If we just take a toy example, something much simpler than what I showed you, we just take 5 places in the brain. So we have some matrix, like what I just showed you, but only for 5 regions in the brain. Some of them are very strongly connected to one another, so they're always on together, like region 3 and 2 here. We have a big red entry, and some of them less, or some of them are anti-correlated. So I get a pattern of which regions go on and off together, how they're functionally connected in this little toy example. I do that for a whole bunch of subjects, thousands often, or hundreds at least, and all I do is I take this pattern, I make it into a nice linear representation here, and I stack it up for 1,000 subjects, and I ask, can I find some model, can I train some machine learning algorithm that would predict from this brain data something across the same 1,000 subjects that's behavioral, in this particular example, attention, but it could be personality or intelligence or anything else about people. So that's the basic idea. You train up a model and you ask, can I predict something about their behavior, and how well does that correlate with the actual measured behavior? So let me tell you about the domain that we looked at. There are many domains you could investigate, but for a number of reasons we focused on these 2. One big reason is something I briefly mentioned at the beginning. Some things you measure about a person are stable, what we call traits. Personality is kind of like that. It's hard to change your personality. It's hard to change your intelligence once you're an adult. On the other hand, your mood you can change. Your behavioral performance on performance tasks you can change. But these seem like relatively stable trait-like variables. And so it would seem reasonable that this equally stable sort of trait-like pattern, this fingerprint that we find in fMRI data, could predict these. It's not something that's momentary and fluctuating, but something where at one point in time I can scan you, and in general I can predict something about your personality and intelligence, because all these things are stable, more or less, over time. And these are the people in my lab who worked on this. A lot of folks, most of the main computational work was done by this guy, Julien Dubois, who's a postdoc in the lab, and lots of other people involved. Lynn Paul is a clinical psychologist. So it's actually a nice team. I should probably say a little bit more about this. This guy's an MR physicist. This person is a clinical psychologist. This guy's a data computer scientist. This guy's a philosopher, faculty at Caltech. This guy's a physicist. This person is a neurobiology grad student, and so forth. So it's a very diverse team that we've been looking at this question. What can we predict from brain data, resting state functional connectivity, if I just put you in a scanner, about what kind of person you are? People have gone through this exercise, of course, in the past, and there are many, many examples. Perhaps the best one, the most studied one, is that there's something very simple about the brain that predicts intelligence, which is just how big your brain is, which it turns out, I was just chatting with David Amaral earlier today, is much more complicated than it sounds. So it turns out big brains can be bad for you, depending, but in general, in healthy individuals, there's a correlation. The larger your brain, the higher your intelligence is. What this shows is all the studies that have reported this, they've just measured the size of your whole brain back from the 1990s, and they get more and more recent, and this shows the effect size of that finding. And what you find is that people tended to report pretty big effects early on, and as time got more and more recent, the effects kind of shrunk and got closer and closer to 0. And if you plotted this, it looks something like this, so that if you look over time, people reported bigger brains are smarter, and they reported big effect sizes at some points in the past. Those tended to gradually get less and less. And this is a general trend that you see throughout this literature, not just for intelligence. And there are basically 3 points to this. One is that in general, you see some kind of regression down towards smaller effect sizes with time. For many of these, they don't hit 0, and it doesn't hit 0 there. So big brains really are correlated with intelligence. It's just not 0.5 or 0.6 or what some of these studies reported earlier. And the other thing that you find is that the circles here, this is the sample size in these studies, tend to get larger as you get more recent. So one problem with most prior studies attempting to answer the kind of question that we did was that in the past, they just weren't very statistically reliable and they wouldn't replicate. The samples were too small and often they had many other problems too. So that's one big caveat, and that was one realization that that's why it took us 2 years to try to do this, because it's actually quite difficult to do properly. So predicting what kind of person you are from resting state fMRI, we analyzed several data sets. The one I'm just going to show you data from is something called the Human Connectome Project. It's a public data set. It's about 1,000 subjects. You have the brain data, and you have lots of measures about these people. Can we use the brain data to predict something about the people? And so one good thing is, so you have 2 data sets, the fMRI, the personality, the Human Connectome Project, 1,000 subjects. We asked a lot of questions, and I won't go through any of these other than just to list the questions for you to give you an idea why it took us some time and why there are still major issues to be resolved with this kind of work. You have to ask what these measures actually measure. I could give you a personality questionnaire, and you could generate any personality you like, depending on how you answer that. So it has to be valid. And there's sort of questions about how good are these measures? Are they actually valid? Is there good retest reliability? So we looked at all of this. They took 2 brain scans, for instance. We looked at both. We asked, is there retest reliability with respect to the brain scans? We looked at the behavioral measures. So we went through all of these different things. And then we did a lot of stuff to figure out how to have clean data that would not mislead us, and how to answer this question of how we could predict something about a person from fMRI data in a way that would generalize, that would be statistically reliable but reproducible and would generalize. And so there's various techniques, cross-validation, permutation testing, all the ones that are listed here, that all required a lot of time, and they would have been impossible to do, well, certainly 10 years ago, maybe even 5 years ago, because they also require a lot of computers to crank through all this data and do all of this. It's possible now, and you will see many more studies of this sort appearing. Most of them in the past have been like the examples I showed you for the correlation between brain size and intelligence, not that well done. Sample sizes are too small, and so there's lots of flaws with prior studies, but we're at the point in time where we have the data and the tools now to do this. And so in the first paper we did, we asked, can we predict aspects of personality from this? There are questions about what personality is, how to measure it. In the Human Connectome data set, they used a fairly well-accepted and psychometrically fairly good measure that characterizes personality across these 5 dimensions that you've probably heard about: openness to experience, extroversion, neuroticism, conscientiousness, and agreeableness. And so there's self-report questionnaires. That's the raw data. It turns out that they have reasonable construct validity, good retest reliability, et cetera. They're fairly heritable, so this just lists the estimated heritability of these personality traits. At least some of these personalities, obviously not based on self-report questionnaires, but on behavior, you can also attribute reliably to animals, to non-human animals as well. So there's reason to think that these are not just made up. There's something real there. And if we actually look at the raw data that we used in the Human Connectome Project data set, it also looked reasonably well behaved and distributed. So this just shows you the actual raw data. This plots the histograms of how many subjects had each score on openness, conscientiousness, extroversion, agreeableness, and neuroticism. And they're like what you would expect given the selection criteria in that data set. People are pretty high on these and they're not neurotic, but that's because they were selected to be that way. So we have those data on the one hand, and we have the brain data on the other, and then we spent a lot of time trying to put the 2 together. One thing, without going into the details, to just point out is there are many, many ways that you can process the fMRI data, many. All of these different steps here, scrubbing, normalization, detrending, blah, 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 are different things that people have to do to the fMRI data to use it at all. But you can choose to do this in different ways, and you can take all these different colored paths through here. And if you add all this up, there are thousands of different ways of analyzing the fMRI data. If you just tried all of them until you found something, you would certainly find something, and it would not replicate. And so we built in a lot of assurances to make sure that what we found would not only replicate, but would be fairly robust with respect to the details of how you did this. So what can we predict? Well, we found that some personality traits, like neuroticism, at least with our data, with our methods, we found no evidence that you could predict this from resting state functional connectivity data. And let me just tell you how this is represented. So all of these little different symbols here are the results, the actual correlation of how well our prediction from the brain data correlates with the actual questionnaire-based measure of neuroticism. So if it was 1, it would be perfect prediction. If it's at 0, it's a chance. And you can see it's pretty close to 0. So one of these here, the REST2 y-axis, is one resting state data set. Each subject had 2. And the other one down here, the x-axis, is a separate, independent resting state data set. What we would like is to find all our results up here showing that they have a pretty big effect size. We can predict with a correlation of 0.3 or 0.4 or whatever it is, both in resting state data set 1, and it replicates in resting state data 2. So this line here, you could think of as sort of line of replicability. This gray shaded area in here corresponds to statistically non-significant results. So for neuroticism, we can really say you didn't find anything. All these different colors and symbols are the different paths that I showed you for analyzing the fMRI data. And you can say they all make some difference. None of them makes it out of this gray shaded box. So we really can't do anything to predict neuroticism from resting state fMRI data. We can predict openness to experience weakly. And so there it looks more like what you would like to find. There's still scatter, so depending on exactly how we choose to analyze the data, it makes a difference. That's the difference between all these different symbols. But they're not wildly different. They're not all over the space. They all cluster up in here, showing that both for this one resting state data set, REST1, and for the second independent resting state data set from the same person, I think these were collected about a week apart, we get correlations of around between 0.1 and 0.2, outside of this gray box. So this gives us some reassurance. We're getting some effect. It's weak. This doesn't account for a large percent of your variance, like 4% or something. But it's giving you something, and it shows some correlation. So these are the kinds of results that we and other people are starting to find. There's substantial room for improvement here, I think. And that's borne out by the second thing that we looked at, which is currently just this paper on bioRxiv, where we tried to predict intelligence. Now, the nice thing there is that you can use different ways to measure intelligence behaviorally. So the way that they do it in the Human Connectome Project data set is by giving you one of these tests. You may have seen these online or taken one of these. The task is, if you get this up there and this, then this is the answer. If you have this first one and this second one, then the third one is the answer. If you have this one and this one, what's the answer? Pick from below. So you start, it's not easy. In fact, I'm not sure what the answer is. But you start, you can see, just knowing geography of the world or knowing facts in the recent news probably won't help you with this. This is an abstract task where you're trying to figure out patterns and rules, so-called fluid intelligence. And so this is what this task indeed measures. And if we use this task that's available then for all the 1,000 subjects in the Human Connectome Project data set, we get something that looks pretty good, looks at least as good or maybe better than in the case of extroversion or openness for personality. So once again, we have these two resting state data sets, fMRI data sets, REST1, REST2. This is the line of reproducibility. This gray area here, the numbers are too small, and this gray area means this is statistically not significant from permutation testing. And we're getting stuff that's statistically significant. You'll notice that there's a separation between the blue and the red symbols. The red symbols are all from a way of parcellating the brain that has more parcels and a more sophisticated way of parcellating the brain from what Matt Glasser and David Van Essen have come up with from the Human Connectome Project data set, whereas these blue ones are a more older way of parcellating the brain, the standard Harvard-Oxford atlas. So it matters also how you define the parcels in the brain. It matters how you process the fMRI data. This makes it hard because you need to test all these different ways of doing things, and then you need to make sure that what you get isn't just a fluke. But you see that there's fairly good reproducibility here, and there's a story here that apparently this more modern parcellation, which has a larger number of parcels, is better for this, is more predictive. It's still not great, so we're still at about 2, a correlation of 2, which in the simplest case you would just square, so it's like 4 or 5% of the variance in predicting intelligence on this task is accounted for by this analysis of the fMRI data. But we can do better, and this is one direction that we've been heading in and doing more work on. The fact is that just taking this Raven's progressive matrices task, or filling out, as it turns out, 12 self-report items on a personality questionnaire, is not a very good measure. In fact, the behavioral measures are typically a lot worse than the brain measure for these data sets. So you can derive intelligence in an alternative way, shown here. This is what Spearman initially had in mind when he came up with this construct G. You can do a factor analysis across all the different tasks. So there are many, many different tasks. We took 10 of them in the Human Connectome Project data set. We have all this information for subjects. And then you do a hierarchical factor analysis that basically says, take all the different performance tasks that people do, attention task, a memory task, a perception task. We have a whole bunch. On average, they're all positively correlated. If you're good at one, you're also good at the others. And that intercorrelation across all the tasks is what this fluid intelligence, or Spearman's G, amounts to. It's pretty well correlated with your score on that Raven's progressive matrices test at about 0.7 or so, but not the same thing. It's a much more precise estimate of this latent variable intelligence than the Raven's progressive matrices task is. And if you do that, you get where the yellow circle is. So you actually get up to about 0.4 or 0.5 in the correlation. So we have reason to think that indeed resting state functional connectivity in the brain is not just a fingerprint from which we can tell you apart from other people, but it's a fingerprint that will predict personality and intelligence. It's difficult. There's lots of processing to do. There's lots of things you have to do to make sure that you get results that are replicable. But the main limitation is in having a good estimate of this latent variable, intelligence or personality, in the first place. And if you just give somebody, a 12-item self-report questionnaire, it's probably a really noisy estimate. So the reason you can't predict personality very well is not because of the brain imaging data, it's because of the behavioral measure. And so that's one main thing that we're working on, coming up with more precise estimates of these constructs, these latent variables, like intelligence or personality. So to summarize this part here, can we predict what kind of person you are from fMRI? The answer is yes, for sure, for some aspects of what kind of person you are, like general intelligence, like Spearman's G. That one works pretty well, and it works pretty well in part because we can just get a more precise measure of it across many, many tasks that people have taken than a single questionnaire would give you. It still requires a large sample of people, so we've done lots of analyses on subsets of these 1,000 patients to get some sense, well, what happens if we only do it on 100 patients or 50? And even in the best case, to get something that's reproducible and fairly stable, you need about 300 or 400 subjects, at least in the work that we've done. And so I think this, right now, if I get papers, if I get studies that estimate to do anything like this, and they only have 100 subjects, that's not going to be enough, at least not yet. You need fairly large samples to build up a predictive model in the first place. And then you have to do it with lots of caveats, and there's still lots of confounds to think about. And so in the closing couple of minutes here, let me tell you where we're going with this and what some of the future implications are of this whole sort of framework that I've shown you. So we know we can predict something about a person from resting state fMRI data. There's lots of caveats, but at least in the best case, we can actually do pretty well. For general intelligence derived as Spearman's G, the correlation is about 0.4 or 0.5. That's quite good. Another question would be, can we predict something categorical about people? So one natural question, of course, is could we predict psychiatric categories? Could we predict whether somebody has autism or not from resting state functional connectivity data? People are doing all of that as well, and it has exactly the same problems that I just mentioned to you. You need large sample sizes, and you need to make sure that what you get would be statistically robust and replicable. And so you can collect data like this from everybody, including people with autism. We did this back in 2014 in this paper. What this shows, this is a bit dense, I apologize, but what this shows is that resting state connectivity matrix in a sample, and I'm blanking now on the number, it wasn't enough, but maybe 30 or something, maybe a bit more, maybe 30 or so. healthy, typically developed individuals matched on lots and lots of variables to a sample, that's this connectivity matrix here, of high-functioning young adults with autism. And you can see if you just compare these, they look about the same. If you subtract them from one another, you get this picture down here. All the green entries here show places in the brain, or pairwise connections between brain regions, where there is a stronger correlation, stronger connectivity in the typically developed brains than in the brains of people with autism. So it is the case that very weakly across the board, the brains of individuals with autism seem to have less functional connectivity on here than do the typically developed brains. However, if you look in detail, if you count across all these rows and you look at particular brain regions, really none of them reach statistical significance, which is this line, with a few exceptions. And so one interesting question, of course, from this is, well, which brain regions do have different connectivity in the brains of people with autism compared to typically developed individuals? And so we're getting data like this, and bigger databases with better data will surely find some differences. It's complicated by another big problem that people are quite aware of, which is even if you get a difference, then you have to ask, well, what is it due to? And is it really due to something in the brain or something that is confounded with or has an effect on the brain imaging data? And so it turns out you can predict things from other data as well that are often confounded with the brain signal. One big one is motion. So if you just take, in our case, if we take people with autism and controls, and we look across this entire big matrix that I just showed you and ask across all the people, what's the histogram of their correlations, you find that autism and typically developed control groups are very similar. That's what I just showed you. It's really hard to see a difference. You can see a little bit of red peeking through here, so there's a tiny difference in that the brains of people with autism have slightly less connectivity, exactly the picture I just showed you, but it's very subtle. However, if I completely disregard that and I just scramble the labels, I take all these subjects together. I no longer distinguish autism from typically developed control, and I simply asked how much did they move when they were in the scanner. And I take those people that moved a lot and those that moved little. I do find differences in connectivity between those groups. So it turns out just how you move, and this could be motion due to how you breathe or how your heart beats, that makes a difference to the functional imaging data. In fact, people have looked at this, and it turns out that to some extent you can predict intelligence by how people move in the scanner. So there's lots of confounds. Motion, your heartbeat, your breathing all contribute to the fMRI signal, and they're also predictive of something about you. So one big challenge is trying to think about all those different measures that might be correlated with the brain imaging data. There are many other measures that we use, and it's worth thinking about which one of these should be the blue ribbon ground truth. Is it the brain imaging data ultimately? Is it, what would it be? And so we tend to think, and I still think, it has to be behavior. So the starting point for any diagnosis is, of course, a description of what people do, how they behave. And it's complicated, it requires consensus amongst clinicians, but I think without that, so I'm not one of those people that thinks that you could just start with a clean slate and just measure stuff from the brain, and we can build up a psychology or a psychiatry from that. I think that's totally impossible. We have to start with the categories we have, and behavior is an interesting category because it's still the ground truth for coming up with these categories. The ultimate goal is to understand complex behavior, and there are differences categorically in behavior, and there are differences in terms of individual differences within the normal range, and those are all these variables that we've been talking about. But it's worth keeping in mind that behavior isn't perfect either. So you can certainly have measurement error, and you can indeed be wrong by looking at behavior. And so one important point to keep in mind is that the flip side of using behavior to make a diagnosis or to judge whether somebody has a certain personality trait is that it builds in stereotypes and biases. So people have looked at this. The most common behavior we'll look at is somebody's face. And so you look at somebody and you can say, the person looks neurotic to me, or the person looks autistic or depressed. Well, people have looked at this in this particular paper. What they did is they got a large sample. It had a lot of faces, and they had people over the internet rate how extrovert, so this is openness to experience, extroversion, agreeableness, and a couple of emotional stability and conscientiousness is what these stand for. How extrovert do you think the person looks? And the end result they came up with is shown in these morphed faces here, that this top row here are the people judged to look like they have the most openness to experience and are the most extrovert, the most agreeable. And as you go down and it continues up here, you get to the least extrovert-looking people. And you can see that one main feature is driving all of this, which is how much they smile. But so this shows that you can get consensus and you can get reliable judgments about what kind of a person somebody is. Alex Todorov, who's a neuroscientist in psychology at Princeton University, recently published this very interesting, nice book, Face Value, where he makes that point. We make judgments about people all the time from looking at their faces, and there's, for some of them, pretty good consensus. Everybody would agree this person looks neurotic, but we're often wrong. So this shows you it's not a good behavioral measure that gives you something about a personality or anything useful about a person. It just reveals our stereotypes and biases. And so this, I think, a key thing to keep in mind. Just very quickly, here's another example of this from a former postdoc of mine, Dan Kennedy, together with Noah Sasson, who did a study recently where they showed people, in this particular plot here, just photos of individuals who either were typically developed or had autism. The people looking at that photos didn't know anything about that. They were simply asked to judge how awkward do you think the person looks, how likely would you want to be friends with this person just from looking at them. And it reveals indeed that there are big biases that correlate with the diagnosis. So all of this to make the point that yes, we need behavior to begin with in order to come up with these categories: personality traits, intelligence, autism, schizophrenia. It's essential. There's no other place to start. On the other hand, you have to be extremely careful because that builds in the potential for biases and stereotypes. If all you're using is how somebody looks, that's probably in the face, that's probably not a good idea. If all you're doing is looking at a couple of photos or a videotape, that's probably insufficient. And that's why it's extremely labor intensive, and we use tools like the ADOS in order to actually triangulate on a diagnosis of autism. It's hard to do. But the hope would be that once we have that ground truth based on detailed behavioral measures, that then if we do find a reliable predictor from fMRI data, it wouldn't have these problems that faces do. It would be a more objective measure, because you can't just look at the functional connectivity matrix and say, oh, the person looks neurotic. It doesn't look like anything. You need to do a lot of processing, and you need to have a very sophisticated model in place. So the hope would be that the brain data would end up being a more objective way to get at the same construct, a personality trait or a psychiatric diagnosis, but it would always be bootstrapped on the ground truth of actual behavior. So what do we want just to wrap up? The hope would be that you doing what we did here, that using functional brain connectivity would not only be a sensitive, a more objective way to predict something about a person, but it would also give you an explanation. Because of course the next step is, if I find that this functional connectivity matrix predicts intelligence, like I showed you, or openness, or whatever it is, the next question is, well, how? What is it in the brain that's driving that prediction? And we can look at that. So we can look in detail at all those little red and blue cells in there, and some of them are not contributing to the prediction, and some of them are. And we can ask, where are they in the brain? Are they in prefrontal cortex, in the amygdala? So you actually get insight that would give you a causal explanation beyond mere prediction. And that would be another reason to use the brain imaging data. It would not just be a biomarker. It would give you insight into an actual causal mechanism. And so the picture would be something like this, that these constructs of personality, intelligence, autism, schizophrenia, all of these derive just from the brain function. So technically, if you were a philosopher, you would say they fully supervene on, or they're constituted by, things happening in the brain. The brain causes behavior, and then there's all these other measures we can use to estimate personality or intelligence by you filling out questionnaires or doing various performance tasks. And that's one set of data we had available in the Human Connectome project. The other set of data we had available was the brain function, the functional connectivity matrix, and everything I showed you asked, can we use this down here, the brain data, to get a prediction of these up here? And the answer was yes, we can do that. But what we ultimately want to then do is have a causal story about particular brain networks. And so we and others are applying lots of tools to go from this, which is just a correlation matrix, to something that has a more stronger causal interpretation of how particular brain regions interact that would allow you to explain personality. So that the final story would be it's some particular influence that one brain region has on another that not only predicts, but explains extroversion or openness or whatever it is. This is a small version of how you would expand this, that ultimately we don't want to stop with the brain, but we want to look at all the distal causes. So in the case of autism, in this sort of very schematized picture here, we would have what we use as the ground truth for the diagnosis, altered behavior, cognition, and emotion. And we know the distal causes are these two, genes and environment and their interaction. And we want to understand everything in between. It's going to be the case that many of these are biomarkers in the sense that they're predictive, like the brain imaging data I showed you. But what we want is to stitch them together so they're not just predictive in isolation, and we have no idea why they're predicting, but we can actually understand how they causally interact with everything to, here's the connectivity part, to cause autism or cause neuroticism or cause changes in intelligence. So, and there's many, many different approaches in here. So one key confound is that people keep publishing these papers. It turns out that there are lots and lots of correlates that you could think of as confounds that you want to control for. And so here is this just sort of for fun, but it turns out in this study, lots of things like the wind speed and the humidity and other things are associated where you live, are associated with personality traits. So there's a huge web. I mean, the phase of the moon when you were born will have some small effect. So there's a huge causal web. And what we want to try is to kind of circumscribe this, to remove all the stuff that's confounds, that's irrelevant, and to kind of hone in on what's an interesting causal mechanism to explain human behavior and cognition, which we tend to think is in the brain. The one place that people have been going, trying to stitch this kind of picture together, is with some of these big data sets. So many of these big data sets, like this UK Biobank one here, that I think eventually is supposed to have 100,000 subjects in it, collects brain imaging data, collects lots of phenotypic data, but also collects genetic data. And so then you can do these analyses, as shown in this paper here, where you can ask, all these different things here are brain measures. So these are different measures of bold signal or diffusion MRI, task MRI measures, resting state fMRI parcellation, blah, blah, blah. And they ask, what's the heritability of all of these brain measures if you just look across all the genes? And they all have some, there's some heritability for all of these. And indeed, you can find, I don't have a picture for that, you can find single SNPs that, in fact, explain variants, explain brain signal in particular brain structures. So that's one direction that people are going in. The other direction is one that we're not going in that direction, but one direction that we've been going in is to do basically the same thing as what I told you, but rather than saying we take healthy individuals and we image their brains and then we want to predict IQ, we take brains that have lesions in them and we ask what effect on IQ does that have. So that's a much more causal story. So this was our first foray into that in this paper, and we've done some more work subsequently, where we had reasonably large sample size, I think it might have been 300 subjects or so, with lesions all over the brain. And we can ask, what's the correlation between the location at which I have a lesion and IQ? And we found indeed that there was a distributed network that fit with some theories, frontoparietal integration theory, that revealed a network within which lesions would impact intelligence. So what we're doing moving forward, again, is trying to use as a sort of discovery the resting state fMRI data in large samples to say, can we predict something about intelligence or personality, derive particular features from that, which parts of the brain are important, and then test that causally and have a causal model. Like, for instance, here, this would predict then that if you lesion a particular region, you should change somebody's personality or intelligence. Just two slides to end on. One thing that we hope to get out of this is, or are getting out of this, is a kind of dimensional space in which we could quantify and visualize everybody as a unique individual. It's a little hard to see maybe here, but so what we've done here, these are the same data I showed you before, people's measures on openness, on extroversion, on neuroticism, and the different colors encode their intelligence in the Human Connectome Project data set. And so you get a cloud that shows certain patterns. And this, I don't know if it's easy to see here, but openness to experience, that personality trait, for instance, is somewhat correlated with intelligence. But if you carry this out with enough variables here in a large enough sample, the picture would be that all of you, would have various abilities, attention and memory and personality and so forth, and you would sort of form a big cloud in this dimensional space. And we could ask, how does that cloud look? Are there people at the outside where pathology would arise? Are there particular groups in here? Are there little clouds out here that would correspond to schizophrenia or depression, et cetera? And I think this, the main challenge here is just having enough measures that we can collect on people to get that kind of rich characterization. The final thing to end on is, so everything I've told you is still built on the idea that we kind of know what we're talking about when we talk about personality or intelligence or autism or schizophrenia, all the things I just mentioned. And we do, to some extent. We use them, we have good behavioral ground truth sort of metrics for them. That doesn't mean that they're immutable and shouldn't be revised. And so although I don't think we can just use a data-driven approach and just use brain data to derive psychology and psychiatry, as I mentioned, I think that's impossible. I do think we can use it to revise psychology and psychiatry. So the hope would be that we won't end up with this, like phrenology did, but we would end up with some revision to the current psychological and psychiatric categories. And indeed, we are starting to do that now. So for example, to just end on a concrete example, for the personality tests in the Human Connectome data set, each of the 5 personality dimensions is measured with 12 items, very short little self-report questionnaire, 60 items total. But you could take all those 60 items and you could ask, can I get subsets of those 60 items that are the best predicted from the functional connectivity brain imaging data, much better than openness that I showed you here. Maybe we get a prediction of 0.6 or 0.7 if I mix and match particular items. And we could then ask, well, what is that? So that would be a kind of neurally derived personality trait that would probably cut across several of those big 5 that we standardly use. So we're trying to do things like that to come up with revised categories and ask the question, to what extent can we revise these psychological categories, and do they look very different from what we had originally? Okay, let me end with that and acknowledge all the different people in the lab. Again, just to highlight a person who did the most work, and it's really, unless you've done it, hard to appreciate how much work it is to do all the nitty-gritty of those papers that are currently on bioRxiv. One is in press now, on predicting personality and intelligence, is this postdoc here, Julien Dubois. And he did it together with a bunch of other programmers, mostly undergraduate students who spent a year or so doing this. Lots of other people in the lab. Extremely important are these folks here. Lynn Paul is the clinical psychologist who was involved with this. Mike Tishka is an MR physicist. Tim Armstrong does a lot of testing, for instance, of the patients with agenesis of the corpus callosum and autism in the lab. So these are all staff that are absolutely essential. I partitioned these people into postdocs, grad students, staff, and alumni here, but they're all equally essential. And thank you to many funding agencies that have, over the years, made this possible. Although that particular story I mentioned to you is unfunded, so we're trying to get funding for it. And then if you want more information, here's the lab website, as well as a few other ones. Thanks.
Thank you. I'm actually going to ask my question, and then I'm going to run around and ask your questions. I'll be a little selfish about that. So it's remarkable work that you're doing. And I'm curious, in looking at, when you're talking about using imaging to predict personality and intelligence, and I assume most of the people were adults?
In this particular Human Connectome Project data set, they're all young adults.
So I'm curious, if you're going to go back and look at development and age-related changes, how well you think that might be a predictor in children, in areas that we know in the brain that are involved in social behavior and personality are changing pretty drastically through that time period in adolescence. How well do you think it would predict?
So I think that is indeed, a next place one wants to go. I mean, the only limitation is having enough data available, but there are databases for kids, and I think this is the next place you would go. I mean, you don't come into the world with a particular IQ or personality. It develops. And so finding precursors and having a story that would eventually map out how the brain function, these personality, intelligence, and the relation between the two changes through development or indeed across the lifespan. The same thing would obtain. Aging would be, the other end of the lifespan would be equally interesting. So to ask how this changes over time, so I can just say that this is the question people should investigate. Maybe you want to investigate it. It's limited just by having high quality data available, but these databases are being set up rapidly. I mean, there's a lot of big consortia putting these data out there, so I think it's just a matter of time before we have enough data. We just sunk our teeth into the one that had the best quality large sample resting state data sets, the Human Connectome project, but there's several others now. So it's a great question, yes. The answer is it will be done. Probably not by me, but by many other people.
Well, I could ask a lot more, but I'm going to open it up to other people, or else we could just sit here and have a conversation. So, does anybody have any other questions?
Thanks, Ralph. I really enjoyed that sort of big picture, inspiring talk. I was wondering if you could say more about, I guess first, whether you thought you would do, 0.5 is impressive already, I think, but for G, but I was thinking whether you could look at those other latent variables, say cognitive control, mentalizing, and add the other data that's already available in the Human Connectome project, so different MR structural measures, diffusion-weighted imaging, and so on, and do even better.
Yes, so yeah, thanks for the question, Erie. It's I'm sure we can do better. I think there are at least 3 questions bundled in here. One is just reducing measurement error in a broad sense, and so if I just have more data, I will be able to do better. If I have longer runs of resting state data, we're doing things now, given that if we're looking at something that's supposed to be a kind of a stable trait, you might think it should be whatever it is that's common between the 2 independent resting state fMRI sessions. And so you could look at what's common and what's not. If it's not shared, then you would toss it out. On the behavioral end, yes, what you mentioned, you could put in everything. And then, of course, if you're just interested in prediction, yes, I can add genetic data and structural MRI data, and we can put everything into the pot. And people are doing that as well. I mean, at the end of the day, Steve Smith did something like this on the Human Connectome project, that you just put in everything, you put in all possible tasks, and you do something that's akin to a canonical correlation, which basically takes 2 highly multivariate data sets and asks, can I come up with some latent construct in the middle that best connects them? And you get something that looks nice. It's not very satisfying, because I think then what you get is something that's very non-specific. And you end up saying there's variance all over stuff in the brain that shows some very broad, swath of variance across all these tasks. And while that might be true, it doesn't give you insight. All it does is maximize, the variance between those two. So that brings me to the third point, which is one that we applied, indeed, to all the data I showed you. It turns out if you ask, if I take IQ, or I take Spearman's G, or I take a personality trait, as I showed you in that black and white, that red and blue, checkerboard pattern across all the different psychological tasks, those are correlated with many other things. And so if I can predict extroversion, well, maybe I can only predict it because it happens to be correlated with intelligence, right? And so what we did here is to actually carefully regress out and remove everything else, the opposite of what you were saying. Take everything else out so that what we get is actually specific to that particular task. And those are all the data that I showed you. But it's a good point. It's kind of a tension between wanting to have maximal predictability, in which case you throw everything in and you don't care how it works, and some kind of explanation rather than mere prediction, in which case you actually want to build in some specificity.
Sure. I guess that comes to my next question, which is how then do you develop that cognitive ontology? I completely agree with you that we need to come up with better latent variables that describe our behavior across a wide range of tasks. So I was wondering if you could sort of even say more about what directions you think we can go to do that.
So I think, people's views diverge on this. And there's some people who would say, everything we currently have, words like attention and memory and emotion, intelligence, personality, you should just get rid of, and you should derive something new from scratch. Another way of thinking about it would be this way. Suppose intelligent aliens who have completely different concepts and language than we do land on Earth. They see humans and other animals doing very complicated things. What psychology would they come up with to explain our behavior? Well, so it's, I think that in our case, not the aliens maybe, we do need to start with what we have. The categories that psychiatry has, the diagnostic categories, the categories that psychology has, and work on refining those rather than just starting with a clean slate. For a number of reasons. One simple one would be that if the aliens land on Earth and they give me some psychology that I cannot at all understand, I don't know that that's very useful. So there may be many different alternative ways of classifying human behavior and variability in it and coming up with categories, including categories of pathological behavior. But if there's somehow, I think the ultimate answer to your question is use, utility. Can we use this for something? Can we understand it? Can we use it for how to behave towards other people? Can we use it for treatment? And then the answer so the full answer to your question may be that there might not be a single psychology or a single psychiatry. You might have somewhat different categories depending on what it is that you're interested in. That might be the eventual result. And I could imagine a future where maybe there is a psychology that scientists use for certain purposes that looks very alien to us and we can't really understand the categories. I'm still going to use personality, intelligence, emotion, memory in everyday life. And so there may be various schemes that are good for various purposes.
Thank you for a wonderful talk, a technical tour de force, and fascinatingly inspiring key into thinking about very large issues. My question concerns what resting state is, and the tension between using an opportunistic current trend in something and the fact that this is an actual uncontrolled task. And when you throw out what doesn't repeat, you have a context that's very powerful, being in a scanner, that may very well repeat. But what we're really looking at is the convolving of the task of being alive and having a sensorium with unknown mental contents and being in a scanner.
Excellent point. Yes, so there are lots of questions about what resting. So the data we have that we used in the Human Connectome project, and they're kind of popular to collect because they're easy to collect. You don't need a task. You just put someone in a scanner, they just lie there, and you collect so-called resting state fMRI. The fact is that, again, it depends, but by and large, it gives you reliable structure. It gives you structure that's reproducible within a subject. People have done this looking at subjects. The guy Russ Poldrack at Stanford scanned himself for a year. And you get reproducible, reliable patterns, not identical. So your resting state functional connectivity pattern isn't identical from one time to another, but it's more similar than to any other people. You find it in infants, you find it when people sleep, and you find it in monkeys. Again, not identical to humans, but you find similar kinds of large-scale network organizations. So I think the first pass answer to your question would be, it depends at sort of what level of detail you look, and that there's some level at a fairly coarse level. So you're not looking at dynamic resting state, and you're maybe not looking at voxel-wise data, but something similar to the matrices that I showed you, that they're coarse patterns that indeed are fairly robust and reliable, and we can use them. And so that seems to be the case. But beyond that, indeed, there is lots and lots more information and structure in resting state that will, of course, depend on what it is that the person is doing, which we don't know very easily. You could imagine improving on this and having questionnaires afterward, collecting additional data like eye tracking if they're doing resting state with their eyes open, to try to get some insight into what people might be doing that's different when they're just lying in a scanner. And you might find, for instance, that people with a diagnosis of autism do something that's different than typically developed individuals. And it's that's responsible for your ability
that's precisely
to
what I'm
use the
pointing
difference
to.
here. Indeed. So that gets back to these sort of causal antecedents and confounds, that they could be breathing differently, they could be thinking about different things, they could be having different brain responses to the sound of the scanner, indeed. And it would be important to try to, if you want to go beyond, all you're interested in is mere prediction, you might not care about any of these. You just say, I can predict it from resting state data. I don't care what the confounds are. But if you actually want an explanation and some causal mechanistic story, then you have to ask exactly the question that you asked, and you have to start experimentally manipulating these. Change the noise of the scanner, change if it's eyes open or eyes closed, and see if it makes a difference. That's important to do, and it gets complicated.
A quick follow-up, which is sort of a rotation of this entire program, would be to take the approach like Gonzalez-Castillo, and you investigate a single brain, but you scan them 100 times. And
Yes,
you get
yes.
extraordinarily statistically reliable single individual data that you then could relate as almost a case study approach.
Right. So people are doing that as well. I mean, there's full range from somebody just scanning their own brain for a year to scanning 10 people or so a bunch of times for many hours, to the Human Connectome project where you have 1,000 subjects and you can't scan each of them for days. See, there was one follow-up to your question. There's another set of data that I think will indeed be better than resting state data that's just emerging, and we're starting to collect it. There are databases now for this, and that's not resting states. You're not just lying in the scanner. It's very uncontrolled, and different people might be doing different things, and you don't know what they're doing. And also in resting state, because you're not really doing anything, if you just look at the brain signal, it's pretty weak. It's not as if you're getting lots of sensory input that's driving regions. You can have people watch movies in the scanner, and this is both more entertaining to them, so they're happier to lie in the scanner watching a movie than do nothing. And it gives you much bigger signal. And if everybody's watching the same movie, you are kind of equating.
It's like Uri Hassan.
Right, Uri Hassan at Princeton, for instance, has done this. Many people are doing this. And they're now well-annotated movies where people have put out big data sets like this. So I think one direction that we'll see in the future is movie-evoked brain imaging data. And you can derive, by the way, the same kinds of fingerprints, the same kinds of networks from movie fMRI as from resting state fMRI. You actually have a lot more signal to work with. So movies give you much more, because from the movies you can actually then back out what it is about the movie that's driving the brain activation. Whenever a face appears in the movie, whenever there's something funny. And so it allows a much easier link going from mere prediction to actual explanation and a causal story.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.